Welcome, everybody. Welcome to this International Day of Immunology. What a time for us to be immunologists. My name is Faith Ossia. I'm the president of the IUIS. I'd like to welcome you on behalf of the IUIS to this special celebration. I think we are all acutely aware of the power of immunology to solve really pertinent problems. The mission of the IUIS is immunology without borders. And now we are facing a pandemic that similarly is without borders. And today we want to reflect on the achievements, the challenges, and the opportunities that COVID-19 has brought to us. So I want to welcome you very, very warmly from wherever you're calling in from. We have participants from in the morning, in the night, in the afternoon, wherever you are, a very, very warm welcome from the IUIS. I will pass over to my co-chair, Andreas, who will also make some introductory comments. Thank you and welcome.
Thank you very much, Faith. It's a great pleasure for me as well. On behalf of the European Federation of Immunological Societies, a subdivision of IUIS, covering about 35 national societies of immunology, about 15,000 immunologists in the area. It's a great pleasure actually to join you here through this day, special day, the Day of Immunology 2021. And we're in the midst of an event that actually shows that pathogens show no borders, they respect no borders, that the pandemic requires international collaboration, but it also shows actually how mighty immunology is in providing vaccination, antigen fast tests, rapid tests, and therapeutic antibodies. We will discuss that in the afternoon. It's a great pleasure for me actually to thank the organizers of this special event. It's, I think, a brainchild of Federica Salustro, and together with Clive Gray, Miriam Mirat, and Pablo Engel, they have set up this program that I hope, I'm pretty sure actually that you will enjoy it.
I've got a special treat for you now. We'll have our opening message by Anthony Fauci. He was appointed director of the US National Institute of Allergy and Infectious Diseases in 1984, and he's since advised 7 presidents on a multitude of domestic and global health issues. He oversees an extensive portfolio of basic and applied research to prevent, diagnose, and treat established infectious disease such as HIV, respiratory infections, diarrheal disease, TB, malaria, and emerging diseases such as Ebola, Zika, and our topic for today, COVID-19. So welcome to this presentation by Anthony Fauci. Thank you.
Thank you for that kind introduction. On this Day of Immunology, it gives me great pleasure to greet my fellow immunologists from around the world gathered to share information regarding COVID-19. Currently, we are living through the most devastating pandemic of a respiratory illness since the historic influenza pandemic of 1918, the defining public health crisis of our grandparents' and great-grandparents' generations. As an immunologist, physician, and U.S. government official who for decades has helped lead the public health response to emerging infectious diseases, it has become patently clear to me the important role that immunology plays in medicine in general and in infectious diseases specifically. It is particularly striking in the COVID-19 challenge we are facing today. For example, the pathogenesis of COVID-19 is correlated with an aberrant immune and inflammatory response to SARS-CoV-2 in infected individuals who ultimately develop advanced disease. People with healthy, intact immune systems, such as most young people, can control SARS-CoV-2 infection, limiting its effects to the upper airways and mild symptoms. In contrast, older people or individuals whose immunity is compromised either by immunosuppressive agents or an immune-compromising illness such as cancer are unlikely to produce a robust immune response that can keep SARS-CoV-2 in check. As a result, the virus can travel into the lungs and elsewhere, become a systemic infection, and paradoxically trigger an abnormal immune response and inflammation, which can lead to respiratory distress, organ failure, and even death. Clearly, the pathogenesis of COVID-19 is directly and indirectly driven largely by the interplay between the host immune system and the coronavirus. Yet immunology also takes center stage in our spectacular success in developing countermeasures to prevent serious COVID-19 disease. Candidate vaccines that were developed and authorized for use in record time are now being deployed to protect people throughout the world. Fundamentally, the development of these vaccines was made possible because of what we have learned about immunology during the past several decades. When the history of this COVID-19 pandemic is written, the discipline of immunology will stand out for its important role both in explaining the remarkably protean manifestations of SARS-CoV-2 infection and in enabling us to identify and exploit vulnerabilities in the virus to develop safe and effective vaccines to thwart its pandemic spread. And so, thank you for your outstanding contributions to this body of research, and please enjoy the rest of the meeting.
A sincere thank you to Anthony Fauci for that opening message. Very inspirational, and we're very, very grateful. It's my pleasure now to introduce our next speaker, Davide Corti from Switzerland. He's the Senior Vice President and leading antibody research at Vir Biotechnology. His team's efforts to date have generated 3 clinical stage monoclonal antibody candidates targeting influenza A, Ebola virus, and HCMV. Welcome, David.
Thank you. Thank you very much. So good morning, good afternoon, good evening, and I think good night to all the immunologists who are connected from all over the world, as I've seen in the chat. So first of all, I would like to thank the organizer for the kind invitation. And I must say that I'm really honored to be part of this panelist and to be presenting after such important presentation before me. So I would like to tell you about overall a success story about a monoclonal antibody that we started developing early on last year. That is an antibody able to cross neutralize multiple viruses of the same family. And this antibody ended up to be successful in the clinic as it was reported recently. Just a second. Okay. So, and I would like to start with acknowledging all the people who have been working on this project from Vir Biotechnology, which is a biotech that has the headquarters in San Francisco, but in other cities in the United States, as well as in Switzerland, where I am in Bellinzona. And I would like to thank Antonio Lansavecchia, that is working with me at UMAPS, that is a subsidiary of Vir, as well as my colleagues in the US, Skip Virgin in particular, and all the other collaborators, including Federica Salusto, who have made possible the success of all this early development of this antibody. So the field of anti-infective monoclonal antibodies has clearly seen an acceleration in the development and approval of monoclonal antibodies against infectious disease target. The first antibody that was approved in this space goes back to 1998, which was palivizumab to prevent severe RSV in premature infants. And that has been followed by a few other antibodies. But there was clearly in the last 2-3 years an incredible acceleration in the approval of antibodies against Ebola in particular, and as we have heard about COVID-19. We have been working, and we are on the Ebola monoclonal antibody and sulimumab, which just at the end of the last year was approved by the FDA to treat Ebola virus disease. The success of the development of monoclonal antibodies to infectious disease target has been largely made possible from the isolation of antibodies out of human memory B cells or plasma cells. So the antibodies were human in origin. And that multiple technologies have been developed to do that, and we have also developed our own platform over the years. And interestingly, the very first set of antibodies that were isolated with this platform goes back to 2004, and the antibodies were directed to SARS-CoV. The isolation of antibodies is not the end of the story. There is a lot of work to be done once the lead antibodies are identified, and this has to do with the engineering of the variable region of the antibody, to improve the developability properties and the manufacturability properties of the identified molecules, but also to improve by the engineering of the constant Fc region, other properties of the antibodies like half-life and effector function. And ultimately, the isolated antibodies can be produced in large scale to be transferred to provide the original protective immunity for which they were generated, in particular to at-risk individuals. So coronaviruses have entered the human population a long time ago. The molecular clock dating put back to the first appearance of a coronavirus into humans back to more than 700 years ago. But clearly in the last 20 years, we have seen more coronaviruses. At least we have been aware of more coronaviruses that have entered the human population, starting from SARS-CoV in 2003, and then several spillover events from dromedaries into humans in the Middle East for MERS-CoV. And we know that SARS-CoV entered the human population towards the end of 2019. And we wanted to take advantage from the fact that SARS-CoV and SARS-CoV-2 do both belong to the same lineage, that is sarbecovirus lineage, even if they originated from independent clades. And they do overall share a 76% homology at the level of the spike. So at the beginning of the pandemic, back to January, February last year, we took advantage on the possibility of isolating antibodies to fight COVID-19, not from COVID-19 patients, but from an individual or individuals that were exposed many years ago to SARS-CoV. And to do so, we isolated from one donor, actually 10 years after infection, a large set of monoclonal antibodies that were found to be cross-reactive between SARS-CoV, against which they were originally generated, but also cross-reactive against SARS-CoV, the spike of SARS-CoV-2. And a lot of these antibodies were found just to bind and to cross-react in terms of binding to the spike protein. But one antibody of this set stood out for its interesting and remarkable neutralizing activity, which was measured against the SARS-CoV and other bat-related SARS coronaviruses, as well as against the SARS-CoV-2. And the activity of this antibody was also confirmed against the authentic SARS-CoV virus. The antibody was found to bind to the spike protein, and in particular to the RBD region within the spike protein with very high affinity, as you can see from this plot showing the on and off rate of the antibody, which is in the range of picomolar affinity. And in collaboration with David Wissler at the University of Washington, we could very fast determine the atomic interaction and the structure of the antibody S309 FAB fragment bound to the both partially open S trimer as well as to the closed trimer. And so we could identify the epitope specificity of the antibody, and also point out that the antibody can recognize both the open and closed states of the spike protein. And here on the right, you can see a nice rendering of the full IgG molecule of the antibody. And as you can see, the antibody itself is not clashing with the binding of the ACE2 receptor that here is shown in red. So we have obviously then investigated further on the mechanism of action of this antibody. But before going into that, I would like just to mention something interesting about the epitope of the antibody, of this antibody, that was found to be a glycopeptide epitope. So it comprises a protein region on the RBD surface, but also a very conserved glycan at position 343 that is extremely conserved within all the viruses of the same lineage, so all sarbecoviruses, and found to be very important for viral fitness, because the genetic removal of this glycan results into a much lower expression of the spike and in a reduced viral infectivity. The antibody is, as you can see, binding to a site on the RBD that is distal from the so-called receptor binding motif, so the footprint of the ACE2 receptor on the RBD. And indeed, as expected, the antibody itself is not interfering with ACE2, directly interfering with ACE2, with the binding of RBD to the ACE2. And in a recent study that we published a few weeks ago in bioRxiv, we could actually demonstrate that the antibody S309 is able to interfere with the fusogenic rearrangement of the spike. And in this specific experiment, we could find out that several antibodies targeting the RBM, the receptor binding motif region, that actually can act as receptor mimics and can trigger a fusogenic rearrangement of the spike, leading to this post-fusion state that is seen here as rosettes, similarly to what the ACE2 receptor does, including, and these antibodies can also promote the formation of syncytia by cells expressing the spike. We could demonstrate that the S309 can actually block this fusogenic rearrangement elicited by the receptor or by this fusogenic monoclonal antibodies. So it seems that the antibody can stabilize the spike protein in a prefusion state. And in the same study, we could also demonstrate that the activity of the antibody is clearly reduced when the ACE2 is overexpressed. And this is a very important technical aspect, because a lot of laboratories are using cells overexpressing ACE2 on the target cells to facilitate the infection. But this is for sure a very good tool for measuring the neutralizing activity of RBM antibodies, but this seems really not to be that much adequate to measure the neutralizing activity of antibodies that are not targeting the receptor binding motif. Another very interesting recent finding is that we found that lectins like L-sign, DC-sign, and also another receptor that we found that is a Siglec-1 can actually enhance the ACE2 mediated dependent entry of SARS-CoV-2. And we found out that antibodies like S309 can very effectively block the lectin facilitated infection. And this is not always the case for antibodies targeting the RBM site. The antibody was also shown to be very effective in a relevant animal model of infection, which is the hamster, in which we could demonstrate in a prophylactic setting. Now we do also have data in a therapeutic setting showing that the antibody can be very effective in reducing the infection of animals to a level that it seems to be comparable in terms of potency to that of receptor binding motif directed antibodies. And the majority, actually not all the other antibodies that have been developed clinically, are all targeting the receptor binding motif. So in that respect, this antibody has some clear unique properties and different mechanisms of action. And amongst those mechanisms of action, we immediately were focused also on the ability of the antibody not just to neutralize viral infectivity, but also to mediate effector function, which in this case has been shown in vitro by demonstrating the ability of this antibody to promote the killing of infected cells by a natural killer cell, as well as the phagocytosis of infected cells by macrophages. And the importance of effector function has been now illustrated in animal models in at least two relevant studies. This is one of the two studies by Michel Nussenzweig and Shinan, showing I think two important aspects. The first is that they have clearly seen that there is a lack of correlation between in vitro neutralization and in vivo potency. And the second important finding is that they see that the activity of some of the antibody is clearly dependent on effector functions in vivo, and this was demonstrated by engineering the antibodies with this GRLR mutation, so to abrogate the effector function of the administered antibody. And another study by Mike Diamond have confirmed this data, and in hamster demonstrated that the effector function seems to be particularly relevant in a therapeutic setting. So for this reason, and also for the reason that still a lot of other groups have been very concerned on the potential detrimental effect of effector functions, the different companies that have developed monoclonal antibodies into the clinic have adopted very different strategies of the FC, of engineering the FC in order to keep the wild type IgG1 format, or to actually do a silencing of the FC by introducing mutations like LALA. Our option was actually to introduce a mutation prolonging the life of the antibody and providing a potential better biodistribution into the mucosal tissues, as well as in a second variant of the antibody, which has now just entered the clinic last week. We have also introduced an additional mutation, and I don't have enough time to present on this specific mutation that is intended to provide what we call a vaccinal effect that we have seen to be relevant for an influenza antibody. I would like just to mention briefly about the variants and the coverage of variants, the variants of concern. It's clear that the variants have now dominated the pandemic, and they have become prevalent overall over the parental virus. In particular, in the last 2 months, now collectively, all these the variants that are listed here on the right do account for more than 90% of the sequenced circulating viruses. And if we look to all the large set of variants of concern or variants of interest as they are defined, we can actually see that they do display a large set of mutations that are located in the RBD, but also outside the RBD. And they actually do share, in most cases, similar mutations at the same positions of the RBD. And the majority of the key mutations that have been found so far are located within the receptor binding motif of the RBD. You can actually see, I don't have the time to go into details here, but the Indian variant is an interesting example of a variant that is now incorporating 2 key escape mutants of the RBM that are the E484, in this case with the Q substitution, as well as the mutation leucine 450 to arginine that is found that has been found in the California variant. In addition to these mutations, another large set of mutations as well as deletions and recently also insertions have been found and described to be in the N-terminal domain region of the spike. And our studies and other studies have demonstrated these mutations to be also relevant escape mutations. And in the case of the California virus, we have recently demonstrated that there is a very new interesting mechanism of viral escape, which has to do with a completely restructuring of the NTD domain. And the 2 possible mechanisms that are most likely accounting for this plasticity of the receptor binding domain, the introduction of all of these mutations, I think has also to do with the immune pressure that has been introduced by herd immunity and also by, of course, by vaccination itself. And we have found, at least in infected individuals in this study that we published last year, that the majority of the antibodies that are produced against the spike are against the RBD. And of those against the RBD, the large majority of the antibodies, this was done by competition study, serological studies, the majority of these antibodies are directed against these sites that we name 1A and 1B that are part of the RBM. So a very high response can obviously be at the basis of some immune pressure on the virus. And I think another very important mechanism has to do also with the possible interspecies spread of the virus. I think we have had the very clear example of minks last year in Denmark, where the virus was actually infecting minks, and some adaptive mutations were introduced in minks, in particular this 453 mutation, and then the virus actually jumped back into humans. So this interspecies ping pong transmission may actually also be responsible and account for part of the virus evolution that will come. Another interesting aspect is also to try to understand whether the degree of mutation and the level of mutations that are introduced and now seen in the variants may somehow parallel what we are being seeing and what we are seeing constantly to occur for influenza virus, where we know very well about the antigenic drift of the virus. So the introduction of mutations is resulting all in time in the reduction of the neutralizing activity of the antibodies that are directed against the parental virus. This is just a specific example of 2 viruses that actually led to the change of the viral strain. in the vaccine. And these are two examples, one related to 290, which is a coronavirus, where a very interesting study by Jesse Bloom has shown, I think, a nice example of what it should be considered as an antigenic drift of the virus. And I'm just, let's say, putting down the concept that this might also be true for a coronavirus, where in this case, for instance, if we compare the neutralizing activity elicited by Vaccinees in Vaccinees to the South African variant as compared to the parental virus, we are clearly experiencing a similar level of reduced neutralizing potency. In terms of coverage of the monoclonal antibodies that are in the clinic, so far we have shown that our antibody that has been recently renamed as sotrovimab has shown a complete coverage of the variants that is absolutely consistent with the epitope specificity. And we are now in the process of testing all these other variants, including the Indian variant, but we do not really foresee all the mutations that are found in those variants to be of concern for our antibody. This is clearly not necessarily the case for other antibodies that were developed even as cocktails. And it's very important to outline that in the cocktail, when one of the two antibodies is knocked out, the cocktail is obviously going to be still active, but it will act in that case as a monotherapy. So this is a summary of the properties of the 7831 antibody, as I said, now renamed sotrovimab. So it works through blocking the viral entry into cells by the neutralizing. Its neutralizing activity can clear the infected cells by recruiting immune system because of the targeting of a conserved region that is shared between different viruses. As I said, the antibody was isolated from a SARS-CoV donor. This antibody has a very high barrier to resistance, and the dose that was tested in the clinic was low because it was tested at 500 milligrams, and we are in the process of trying to transition the dosing of the antibody into an intramuscular route. And the antibody was also half-life extended with the mutation that I mentioned that is also result into an improved lung bioavailability. The phase 3 study that has been terminated because of overwhelming efficacy a month ago showed that the antibody, sotrovimab antibody, can reduce by 85% the risk of the level of hospitalization or death in high-risk adult outpatients as compared to a placebo. So far, the antibody has shown a very good safety profile from this study, and now we are seeking the emergency use authorization from the FDA. And the antibody was also tested in collaboration with Lilly in the BLAZE-4 study in combination with the Lilly lamivimab antibody. In this case, showing a 70% relative reduction in the persistent high viral load on day 7. So the antibody is going to be possibly approved to be used in these patients. And I think that, and I'm close to the end of my presentation, I think monoclonal antibodies as a bulk, they do represent an important add to vaccines in the armamentariums of therapies in particular, and so far have clearly shown to be effective in the early treatment setting. The studies so far have not shown the antibodies to work in the late treatment, so in particular in hospitalized patients. But I think that more studies are warranted to investigate whether combination of antibodies with maybe other antivirals or anti-inflammatory approaches may actually provide some benefit for the use of antibodies also in that space. I think all in all, there has been a proof now, and I'm quite sure that this will change the field, that antibodies can be rapidly isolated for infectious disease target and for emerging pathogen, and can also be developed very rapidly. And I think that this may change in the future also with the idea of isolating and developing antibodies for potentially emerging new emerging pathogens by targeting conserved regions on those pathogens. I think it's also important to highlight on the relevance of reprogramming effector functions and half-life that can actually result in better activity of the antibodies. And the new technologies have actually allowed to go through a very fast manufacturing of the antibodies, which is definitely unprecedented, because in just 3-4 months the antibodies were produced and deployed to people and delivered to people. And it is now in the process, also under investigation, the possibility of using different delivery methods like DNA, RNAs, and adeno-associated vectors. And we are also working in this space. And to conclude, I think that definitely the antibodies, monoclonal antibodies for COVID-19, but also for other pathogens, they do represent a very important complement to the existing vaccines that are in development for post-exposure and therapy, but also, and this is also under investigation by AstraZeneca, for prophylaxis in at-risk individuals. And with that, I'm finished with my presentation. So we'll be very glad to take questions.
Thank you very much, David. That was really very interesting and very exciting. I think you've actually answered some of the questions, especially with this last slide. And some of those questions were, how do we place monoclonal antibodies in the current landscape of vaccines? Do you see them as prophylactic agents or as therapeutic agents? When you consider the cost and the time, you know, I think you've thought about all those things. Essentially, how do you compete with the other vaccines that are there? I mean, I think we're in a current pandemic where there's also a lot of shortage. So, you know, everything, you could argue everything is needed. Where do you place yourself? Yeah.
I don't really think that it is a matter of competition here. It is really a matter of having additional countermeasures for, as I said, for early therapy, where there is a strong evidence that monoclonal antibodies can work very well. And by reducing the hospitalization rate, obviously that can result into a very significant benefit for the care system. And in terms of the prophylactic approach, obviously that is in direct competition with vaccines, but we should not forget that there are some categories like immunocompromised individuals, like people undergoing chemotherapy, who may be obviously not responding well to vaccinations, and so the passive administration of antibodies to these categories may represent a useful approach for monoclonal antibodies that, through these technologies, as I mentioned, for half-life extension, can be given as a single shot, intramuscular shot, and potentially provide protection, which is what AstraZeneca is looking at for a period that can go from 6 months to a year. So similarly to a vaccine.
And then another general question, thank you for that, is side effects. What are the major side effects you see? What are the risks with antibodies, antibody-dependent enhancement of disease, for example? Could you address that briefly?
Yes. I mean, so far, at least for our own study, and we have also run a study that was named ACTIV-3 in collaboration with the NIH, where the antibody was given to hospitalized patients. We have not seen signs of ADE in people. And that, I think, is also true for the other monoclonal antibodies that have been tested in this setting. So I would say that in a nutshell, there is no proof from the clinical data available so far that ADE seems to be a concern. For some of the antibodies tested in hospitalized patients, they have had some safety signals, but they were not clearly associated to an ADE mechanism, at least so far. And I think it's important that to highlight that not necessarily all the antibodies are the same. So I think more has to be understood in this space, and the monitoring of the safety is very important to rule out any potential concern. But so far, I think that there is nothing that has raised any clear red flag for that.
Okay, and just the last one, thank you, is you showed us nicely all the different variants, and have you considered how you could have a pan-variant monoclonal antibody? Can you predict and design this ahead of time, for example?
Well, I think to some extent we will consider our current antibody pretty much like an antibody like that, because we are targeting a region that is clearly not evolving in the virus, most likely because it is not the immune suppression of of the antibody response. We are looking into other classes of antibodies, including antibodies that are able to cope with the overall diversity of all beta coronaviruses. And we have found such antibody which actually targets the so-called stem helix region of the spike, that I think is a very interesting target for therapeutics, but in my opinion, also for a pan-coronavirus type of vaccine that is very, I think, a very interesting kind of vaccine to be designed, and we are working actively on that in collaboration with GSK.
Fantastic. Thank you very much.
Thank
you. We really appreciate that talk. I can see a very strong hint that we need to move on with the program. So I hand over to Andreas. Thank you, Andreas.
Thank you, Faith. And it's a great pleasure for me to introduce the next speaker, Shane Crotty. Shane actually received his education at the MIT in Boston and then finished his PhD at the University of California in San Francisco, already in virology at that time. And then he joined actually Rafi Ahmed for a postdoc, and that really got him into analyzing the immune response to viruses. From Rafi, actually, he went back to the La Jolla Institute of Immunology, which he joined actually in 2004. And since then, you have risen the ranks of academic career there and done extremely well. The claim to fame of this follicular helper cell biology definition. I still remember when we first met Shane. I arrived after 18 hours of flight in San Diego, and we had dinner together, and I was completely jet lagged. We didn't really have a good discussion, I think. This was my fault. But today I'm actually in a good shape. It's early afternoon, and it's very early for you, and I'm looking forward actually how you will take that challenge. I'm sure you will master it. I'm looking forward actually to this talk on the immune response to SARS-CoV-2. And he and Alessandro Sette published the first successful analysis of this. And in particular, I'm actually looking forward to the data on how long it lasts, actually. So we have heard already quite a bit about that there are T cells and B cells and so on, but the question on the maintenance of immunological memory has not been addressed so far, and Shane will do so. Thank you very much for agreeing to speak to us in this early hour for you.
Thanks, Andreas. Happy Immunology Day. Congrats to you and all the organizers for putting this together. This is a really fun event. I think it's a great idea, and it's been outstanding to see the presentations by Dr. Teresi on her whole BioNTech team RNA vaccine successes, and Corti's amazing monoclonal antibody work. So yeah, it is a little early in the morning here in California, but you know that's part of the nature of trying to do international stuff, right? So I actually got up extra early and biked into work to make sure I'd be awake for it all. So so my charge today is to, as Andreas said, as Dr. Redbrook said, talk some about what do we understand about immune responses to SARS-CoV-2 in the context of natural infection, with particular interest in adaptive immunity. As he mentioned, well, all of the work that I've done has been in close collaboration with Alex Setti here at LGI. It's been a fantastic collaboration between our labs and several other labs. And as he mentioned, we had one of the first big studies to try and measure the T cell and antibody responses, the adaptive immune responses at the same time to this virus, to try and understand the infection. So here's actually the front page of that paper, or if you prefer the live action shot. Here's a photo of Tony Fauci holding up a copy of our paper in Congress. So that study got a lot of attention because there was a lot of fear early in the pandemic about whether people made immune responses to this new virus at all, And if when they did make those immune responses, they did develop immunity to COVID-19. Understanding protective immunity to a virus is usually a very long process, takes quite a few years to really understand something like that in full, particularly in humans. So we are definitely not there yet with that understanding, and maybe that'll be an excellent topic of conversation for the immunology panelists. But you have to start somewhere, and we realized we could plausibly get a good first sense of this, first estimation of the nature of immunity to this disease and SARS-2 infection by studying average cases, that is to say non-hospitalized cases, because this is generally an acute infection that resolves or cures. So average cases probably reflect people who get infected by the virus, have a significant viral load, but then make an adaptive immune response that controls and clears that infection. And so to get at what kinds of immunity are important for controlling and clearing this disease, by looking at immune responses in people who did successfully control the infection, we can get insights into that. And so we felt it was important to measure antibodies, virus-specific CD4s, and virus-specific CD8s all at the same time in the same people, because each of these branches of adaptive immunity have been shown in the past to be important for controlling different viral infections. And sometimes it's all about one of them and sometimes all about the other. But frequently, actually, there's a lot of compensation between these different branches of the immune system, because that's really what they're built to do. Antibodies are important in almost all currently licensed human vaccines. Cd8 T cells are critical for antibody responses, almost all neutralizing antibody responses, but they also have a diversity of functions. They're not, you really can't pigeonhole them in a simple way, including antiviral activity. And it's notable for coronaviruses that Stanley Perlman showed 6 years ago now, that CD4 T cells can actually protect against coronavirus infections, classic SARS, in a mouse model independent of antibodies. And CD8 T cells are clearly important in many viral infections, and really an oversimplified view of immunology is that antibodies are important for stopping viruses outside of cells, and CD8 T cells are important for stopping viruses inside of cells once you've actually had an infection start. And so we did this study to try and establish a benchmark of these immune responses and what protective immunity may entail. And to measure T cell responses can be quite challenging, particularly to larger viruses, and coronaviruses are really the largest of the RNA viruses, because it can be difficult to predict and identify T cell epitopes, and the CD4 and CD8 epitopes are independent. And luckily here at LJI, we have Professor Alex Cedi, a great colleague of mine, who's one of the top experts in the world at predicting and identifying T cell epitopes, particularly in humans. And so we made use of his predicted, we worked together to use their predicted epitopes, class 1 and class 2. And we looked at 20 individuals who had recovered from, again, average cases of COVID-19. And what we observed was really generally considered good news, that all of those individuals made antibody responses against spike, the S protein, but also 100% of people made measurable CD4 responses and 70% made measurable CD8 responses. And the magnitudes of those responses were quite reasonable, we considered. And the phenotypes were also look like good functional T cell responses. And it also is notable that these responses overall were not particularly large. And so the magnitude of those responses suggested that this was not a virus that was particularly challenging for the adaptive immune system to control and clear, okay, at a first pass. And this was in April of last year. And so that was generally considered a good sign for the potential that the vaccines against this virus may not be particularly challenging to develop. One other vaccine-related topic that we looked at the time, because again, this was well before there were any human trial results, was that almost all the vaccines were focused exclusively on spike, and that could be a problem if spike wasn't a good T cell target. And so we were very keen on that. And what we observed was actually spike in the context of natural infection is actually a very good target of CD4 T cells and a good target of Cd8 T cells. And so that was an encouraging sign that if one was going to choose a single antigen vaccine, spike was actually a very reasonable choice, and it was plausible to get decent T cell responses to it. From a viral immunology perspective, it was also good news that the T cell responses were not narrow. The T cell responses were actually quite broad and responded to quite a range of antigens, which is generally an indication of viral immunology of T cell responses that are difficult for viruses to escape. And we can come back to that. In a second study, we shifted to looking at immune responses during acute COVID-19. This is a much more challenging situation to obtain the samples and match the samples. This is a study of 50 individuals, 25 acute, 25 convalescent. And again, our philosophy in this study was that we felt it was important to measure the virus-specific CD8, CD4, and antibody responses all in the same individuals at the same time. And the one-slide summary of that is shown here, where when looking at all of those immune responses at the same time, as well as innate immune responses and disease severities, what we observed was that better outcomes were actually associated with the presence, statistically associated with the presence of virus-specific CD4 responses and virus-specific CD8 responses. And we did not observe a correlation with the antibody responses, which overall we have taken to mean that the coordinated adaptive immunity to this virus is protective immunity. It looks like CD8, CD4s, and antibodies all play roles, can all play roles in helping to control a SARS-CoV-2 infection. But that based on the statistical associations in natural infection, it appears that the T cells do a lot of the heavy lifting in controlling this virus. And in this study, we observed no convincing evidence of likely causal negative associations of adaptive immunity with disease severity. In contrast, on the contrary, actually, severe disease was largely associated with a failure to make a well-coordinated adaptive immune response. It's well known that age is a major COVID-19 risk factor, if not the biggest global COVID-19 risk factor. And while it doesn't have to have anything to do with adaptive immunity, our data indicated that adaptive immune shortcomings are part of the problem of advanced age and COVID-19, potentially due to poorly coordinated T cell responses that in a simple model may come from the fact that individuals over the age of 65 or 75 have a more limited naive T cell repertoire that's going to have to be the source of the majority of the T cell response to a new virus. Okay. For a very important and broad topic of conversation, right, are what are the mechanisms of protective immunity against this disease? From a vaccine perspective, really the simplest option for any vaccine development is that high-level, long-lasting neutralizing antibodies are your best possible option for any pathogen that is susceptible to neutralizing antibodies. And this virus is clearly susceptible to neutralizing antibodies. One of our important considerations as we've looked at immune responses in natural infection have been to try and look at that with an eye towards if one doesn't see high levels of neutralizing antibodies, which, for example, is the case for many people who do have natural infection, the antibody responses are not particularly high, or if the antibody responses are not long-lasting, either for natural immunity or for some vaccines, or, for example, if the neutralizing antibody levels aren't high because variants may obviate some of that potency. Are there other things the human immune system can do to provide protective immunity beyond neutralizing antibodies? Okay. And we think various lines of evidence point to substantial protective contributions of T cells against COVID-19, not necessarily protection from infection itself, but there are many levels of protective immunity, starting with sterilizing immunity of preventing infection, all the way down to protective immunity being prevention of death or prevention of hospitalization, which is certainly a major public health burden. And so our operating model is really based on all the available data. We think it's quite reasonable to consider that serious COVID-19 is prevented by any decent combination of antibodies and or CD4 and or Cd8 T cells. That this virus is sensitive to immune control by each of those branches of adaptive immunity, at least to some degree. And of course, location. The immune response does have to exhibit its effector functions at the sites of infection. And so I just will note that this is clearly an infection of the nasal cavity with an oral component. And the virus is very fast in the nasal cavity, but it's rather slow in the lungs. And to date, most of the immune responses that we've measured, the T cells and the antibodies across the field, measurements in blood have generally correlated with measurements in lung and the oral cavity. So we do think blood measurements are a reasonable reflection of what's going on, but it's always going to be important to get more data at the actual sites of infection so we have a better sense of these factors. So with all of those things in mind, we then turned our attention to immune memory. And this will be the final component that I talk about. And this has been an effort to try and get at the question of, does protective immunity against reinfection occur? And if so, how long does that protective immunity last? And there have been a number of excellent, there's a direct approach to answer that question, which is to take a large cohort of people and track those individuals and wait and observe reinfections over time. And that's a very powerful approach, and many groups are following that. I'll come back to that at the end. And what we took was what we felt was the complementary approach of saying, well, any protection against reinfection will be because of the presence of immunological memory. And so what we did in this study was to measure the components of immunological memory that may contribute to that protective immunity, to try and get a sense of magnitudes and the kinetics of those responses over time. And given that we feel like there's a reasonable likelihood that multiple different components of adaptive immunity could contribute to protection at some level, we again felt it was important to measure these different antigen-specific branches of adaptive immunity at the same time in the same people. So we measured antibodies, but also virus-specific CD4s and virus-specific CD8s over time in people, and additionally memory B cells. So memory B cells don't actively produce antibody, but they can reactivate and make antibody upon reinfection. And so we measured all four of these components, and we did so in 188 individuals, which to the best of our knowledge is the largest study ever for any viral infection to measure these four components of adaptive immunity over this kind of time window. And the additional philosophical point from us has been, we've studied immunological memory for the past 20 years, and painfully it's very difficult to predict immunological memory. And so we've generally felt like it's important to wait 6 months, and it's important to get that kind of time scale of data to then be able to predict and assess what the immune memory looks like. So we did that in this study, and we observed that antibody responses were durable. There are much larger serological studies than ours, including, and really the two that I tend to point out are the studies of thousands of individuals by Florian Kramer and his colleagues at Mount Sinai, and then the Iceland seroprevalence study. Both of those studies have looked at sort of 6-month windows of time and saw results similar to ours, that almost everybody's seropositive, and it's still 90+% out at 6 to 8 months. This is the cross-sectional data. This is the longitudinal data, which we had for 40 to 50 people with 2 to 3 time points as a sanity check that our cross-sectional data was reasonable. And what we were adding to those serological findings were, again, the cellular responses. And so memory B cells, we looked at by flow cytometry, looking for spike-specific, RBD-specific, and nucleocapsid-specific responses. These data really looked quite clean. And these were probably the most surprising findings of the study overall, which is that individuals have more memory B cells at 6 months post-infection than they do at 1 month post-infection. And that was true for spike, RBD, and nucleocapsid, also true for longitudinal data sets, and even clearer if we specifically looked at IgG positive spike memory over time. This has been corroborated by a number of other groups at this point with related publications on memory B cells, and it also appears to not be unique to COVID-19. Which I can come back to. There is IgM memory, but interestingly, it appears to fade relatively rapidly over time. So memory B cell memory is clearly present in most individuals after COVID-19 infection. What about memory Cd8 T cells? These data we could see memory Cd8 T cells in about 70% of individuals at early time points, consistent with our earlier Cell paper. And then at later time points, it was about 50% with a half-life of about 4 months. So a relatively slow decline, and the rate of that decline appears to be pretty similar to the yellow fever vaccine, which is a quite good vaccine. So that was an encouraging sign about this memory. We looked at CD4 memory. Memory CD4s are really quite robust to this viral infection, both in terms of magnitude and percent detectable. And with a decay fairly similar to the CD8s over time. And here's the spike-specific data as well. We did measure memory TFH cells, which are important for helping B cells, and memory TFH cells are also present over time, both for spike and non-spike, interestingly with an enrichment for CCR6 over time. And so if we take all of that data and look at it together, I think there are 2 general things to note. One, there is immunological memory over time after a COVID-19 infection in the majority of individuals, but that immune memory is complex and that there's a lot of heterogeneity from person to person. And so briefly trying to deal with those 2 points, what we did was trying to relate immunological memory to protective immunity. We said, well, we don't know for sure what components of memory are providing that, but we could put forward an educated guess that these 5 components of immune memory are probably the most important. And so then we asked, how do those immune memory components look like in these populations that we studied? And what we observed was that at early time points, most people actually had all 5 of these components of immunological memory. But out at 6 months, where we were more interested, we saw that still 95% of people had at least 3 out of 5 of these immunological memory components, which we interpret to mean that while that amount of memory may not prevent infection in all of those individuals, it is likely to be sufficient immunological memory to be protective against serious reinfection with COVID-19, probably for years into the future based on the kinetics of the immune memory that we were observing. However, there is substantial heterogeneity in the population, and some individuals, approximately 5%, have rather little immune memory for unknown reasons. And perhaps those individuals are more susceptible to reinfection more quickly. Okay, so here's a summary slide of what I just said. And then at the same time we published this, there are really now complementary studies that have come out of directly measuring protective immunity by tracking cohorts of people over time, and really with really quite complementary findings that really large studies in the UK finding substantial protection against reinfection out to 6+ months, and most of the infections observed were asymptomatic. This has come back. It's got a lot of coverage, but it's come back more recently, at least in the US, for arguments about immune memory and COVID infections and wearing masks. And there was a rather fiery debate between one of our senators and Tony Fauci a few weeks ago. I have a full discussion of this on YouTube. I will say that basically one aspect of that argument was that it was important to point out there's heterogeneity between people. And so at a population level, one can't predict who has protective immunity and who doesn't. And then second, that really variants are a big issue for considering reinfections. And I love this article in Scientific American. That's a really nice summary that your immune system has really evolved to fight coronavirus infections. Variants, because it's not just about antibodies. Antibodies are fantastic, but memory B cells can be a source of recognition of variants. And the T cells are very good at recognizing variants. Both the CD4s and CD8s in COVID-19 target a broad spectrum of epitopes, and thus Alex Cedi and his team have shown the variants really are not escaping CD4 and CD8 T cell responses. And based on this concept of protective immunity, we think that, you know, as you get more and more variant divergence, it may become more and a much larger percent of the protection that is observed may be dependent on these other three components, which may be a discussion point for us all later. So I'll stop there. There are a lot of people to thank. Albo Grafoni was an amazing postdoc who led the first cell paper. Jen Dan was responsible for all of the serology, Sidney Ramirez for recruiting the acute subjects, and Carolyn Modebacher was the postdoc who led our acute study. And Assistant Professor Daniela Weisskopf did an amazing job as a co-corresponding author on the immune memory study. So I will stop there, Andreas.
Yeah, thank you very much. Great talk. And will try to distill a few questions from.
It's a broad topic, right? So it's good that there will be the panelist discussion later.
Yeah. So one of maybe a few people asked actually whether you could comment on what makes SARS-CoV-2 so pathogenic for some people but not for others, and discriminates actually SARS-CoV-2 from other coronaviruses, which apparently do not have this potential to become so pathogenic for some people. Yeah. So is there any speculation on what failure of the immune system and how the immune system fails in these people who are intensive care unit patients?
Yeah, it's a fantastic question. Certainly one of the biggest, I'd say, mysteries of the virus has been the massive heterogeneity in the disease outcomes. I think, well, yeah, I mean, if I can show the slide again, we generally talk about it in this way. Oh, do I have the full slide of it? No, I just have the short version. I think there are a number of possibilities. One is that it's simply a new virus in the population, and so there's more heterogeneity in that wave, and we would have observed that with other coronaviruses if we were measuring them at the time they first entered the human population. But to me, the model we've put together to try and explain the heterogeneity is a quite simple model that largely tracks back to the triggering of the innate immune response. To me, any virus that can cause disease in humans has to have at least one good immune evasion trick. And it seems that the big trick, if you will, of SARS-CoV-2 is that it is unusually stealthy at avoiding triggering an early innate immune response. Normally, you would have an early innate immune response very fast, and instead, the type 1, type 3 interferon response is relatively slow to this virus. And that early stealthiness has a lot of trickle-down effects. It gives the virus a head start in the nasal cavities, but it also results in a delay in the adaptive immune response because your adaptive immune response is largely dependent on the innate immune response. So any delay here is likely to cause a delay here. And people with severe, right, hospitalization level disease or fatal disease may be this same process happening, but even further delayed. okay? That if really your innate immune response occurs just too late, then it's going to have your T cell response occurring too late. And the other parameter that comes into play there is that, with that delay, if there isn't enough of a T cell response and antibody response by this point in time, the immune system may essentially be forced to escalate the innate immune response to fill the vacuum left behind by the absence of the adaptive immune response. And really severe disease is largely characterized by massive amounts of neutrophils and myeloid cells, other innate cells in the lung. So that's a simplistic model that I do think accounts for much of the heterogeneity observed and may help explain age differences, right, based on innate immunity. And it's definitely consistent with the work of Casanova and others that people with innate defects are at high risk for severe disease. So it was a long answer, but it's a big question.
Yeah.
Oh, and it also helps explain, I think, the successes of the vaccines, because if it is a race, right, if the whole problem with this virus is that it's a race and the heterogeneity is largely about a race, that's the brilliance of vaccines, is that they get rid of the race. You have the immune response in advance. And so I do think that is consistent with the extraordinary success of the vaccines.
So just one more quick question, quick answer. Several people ask about the value actually of mucosal antibodies. So that is nasal and also fecal IgA, dimeric IgA versus systemic antibodies like IgG and monomeric IgA.
Yeah, I think it's an important topic, and it's one that can only be directly addressed by measuring nasal antibodies. Really, almost all the available data have been mucosal antibodies have been oral antibodies in saliva, and the saliva data have really largely corroborated the blood data that the amounts of IgA and IgG in presence in saliva, the bloods are really, it's a good reflection of that. Whether blood's a good reflection of the nasal cavity remains undefined. Yeah, great questions. Thanks, Andreas.
Thank you, Shane, for this splendid presentation, and I get an unmistakable signal that I should hand over to Faith.
Yes.
Thank you to all our speakers. We've had a really fantastic session. We're going to take a short break now, just a 5-minute break so you can stretch your legs, and then we'll be back for a panel discussion in 5 minutes' time. Thank you very much.
Cloud, you're there. See, I'm here. Okay.
Immunology is a part of scientific field that studies everything about immune system. But for me personally, immunology is a key. It's a key for solving many health and social problems such as aging, cancer, autoimmunity, obesity, and even pandemic.
So for me, immunology is my world. It's the basis of my research, and most important, it's what I enjoy and I love. So as an immunologist, I think that we play a vital role in so many aspects. For example, the development and testing of new vaccines, or creating new treatments that can manage or cure conditions to improve the quality of life of many people.
Immunology research done here in Nairobi has helped us identify a group of sex workers who are naturally resistant to HIV. Understanding this natural resistance to HIV is important in informing the design of effective prevention tools.
To me, immunology is about understanding the cellular superheroes that live inside your body, called immune cells. They're kind of like the Avengers, where there's different types of immune cells and they all have their functions and powers. Immunology is a puzzle, and yet an exciting one.
It's a very complex organization of different cell types and different factors that are interacting with each other to induce a proper immune response. These interactions are so finely tuned that little change, a bit more of these cells or a bit less of this factor, can radically change the immune response in one way or the other.
It's a patient, and immunology is like a medieval story about heroes giving their lives to protect us from microbial invaders and also double agents, I mean cancer cells.
There have been many humanitarian crises during the COVID-19. The best example is COVID-19 vaccine stockpiling by economically strong countries where many countries cannot afford it. In this situation, India has played an important role in production and distribution of vaccines at low cost.
For me, immunology is really one of the most fascinating and interesting fields of biology and medical sciences. The immune system is not just there to protect us from infections, it's really much, much more. And it's just amazing how many different cell types there are that execute different functions in different kind of contexts, and how everything is just super precisely controlled. So I think immunology is just fascinating.
Joe, you can start already.
Sounds good. Hi, everybody. Thanks again for joining us today. I am João Monteiro. I'm the Chief Editor at Nature Medicine. And I would like to welcome everybody to this final part of today's webinar. We will now have a panel discussion with 4 rounds of questions focusing on immunology in the time of COVID-19 and talking a little bit about the achievements and challenges and opportunities for the future in this pandemic and in future ones. Mediating this discussion with me are Nadia Bakuchevich, who is an editor at the European Journal of Immunology based in Frankfurt, Germany. Mia Malan, who is the editor-in-chief of the Bikesisa Center for Health Journalism based in South Africa. And Seth Skellen, who is the immunology editor at Science based in the UK. We are being joined today by an incredible roster of experts from all around the globe who have been actively involved in the response to COVID-19 pandemic and who we warmly welcome to our panel today. Thanks everybody for agreeing to contribute to this discussion. There's a lot to talk about, so I think we should get started, and I will kick it off asking you guys to comment on what the current situation is in your own country in terms of vaccine rollouts and the overall control of the pandemic. And I thought we could start maybe with Alberto Mantovani, who is a past president of the IUIS and is currently the scientific director of the Humanitas Research Institute and a very prolific researcher studying innate immunity in many different contexts, and also in the context now of COVID-19. So Alberto, I know that we all know that the situation in Italy in the very beginning of the pandemic was quite devastating, and things have gotten better. So perhaps you can comment and update us what the current situation is, and what do you think as an immunologist, or where should our focus be on right now in terms of both therapy and understanding the biology of the disease?
Italy was the first Western country to be hardly hit by the virus, and early on, I mean, we had a big question mark. The apparent lethality was much higher than in China. So is there something wrong genetically with Italians, or in particular with people in northern Italy? So that paved the way to our involvement in genetics. I will briefly comment on that. The current situation is that we are easing the lockdown. Eventually, the vaccine program is running. We are vaccinated at a rate. We are going to reach in a couple of days 500,000 vaccinations a day using both the mRNA vaccines and the adeno platform. vaccines. And we made some contribution, and Florian is here, and we have a JCI paper, and Florian and us made what I think is an important contribution, a single dose for patients, for subjects who had experienced COVID-19, which means saving more than 100 million doses of mRNA vaccines worldwide. The genetics, let me just, Shane Crotty mentioned innate immunity as key, but nobody has touched upon it. So genetics, so we did our first study in genetics, and then we have been part of networks. Message is very clear. Genetic polymorphisms that involved in innate immunity pathways, including interferon, chemokines, are key. So that's message number one. Open questions. Let me formulate 3 or 4 open questions. What does the innate immune system recognize? We assume many things. We have a paper which hopefully we'll get through concerning recognition of viral components by innate immunity, but I think it's still a question mark. My second question mark, of course, we have a yin-yang relationship, as again Shane said, early on, late. There is work published in the journal, in your journal, by Miriam, for instance, as an example, Miriam Merad, cytokine chemokine signatures being prognostic. We have our own story on a PET molecule of ours, PTX3, being a very strong prognostic indicator. But I think that we have a key general question, moving from prognosis to prediction, and moving from prognosis to prediction and personalized medicine based on biomarkers and genetics. I think this is one of the challenges that we have in terms of treatment. Final challenge, we all use acute phase proteins or acute phase markers, ferritin and so on, but we really don't understand their significance. So humoral innate immunity, including complement, fluid phase, pattern recognition molecules, antibodies, if you pass me the definition, are still a question mark in terms of prognostic, predictive, and functional significance in the disease. So these are my open questions. I think I stayed in my 2 minutes.
Fantastic. So let me follow up on this issue of understanding the effect of the pandemic and of the disease in different populations in different places. And I'd like to talk now both with Joanna Kierman, who is the Director of the Health Sciences first year and the Professor of Microbiology and Immunology at the University of Otago. She's been advising the New Zealand government on COVID vaccines and strategy over the past year and a half. And also Ron Balicher from Israel, who's a public health physician and also an advisor to the Israeli government. I think both Israel and New Zealand have been seen, from a while now as leading countries in terms of the way your countries responded to the pandemic, and not just in the beginning in terms of implementing NPIs, but also in terms of rolling out vaccinations and really keeping the virus at bay. So maybe Joanna, maybe you can start telling us a little bit about, what was the root for the success of the approach in your country, and what you see now as the major challenge going forward in this second year of the pandemic?
Thanks, Joe. So New Zealand has really been in a fortunate position where we've got geographical isolation, but we've also had our government has been very science-led and really has taken advice on board, which has been great. And very early on in the pandemic, the approach was go early, go hard, and we went into quite a serious lockdown. And during that time, not only did that help to mitigate transmission in New Zealand, but we were able to scale up our infrastructure for testing, have contact tracing infrastructure established. We also, the government was really good at communicating with the public and getting the public on board, not only with the physical distancing and lockdown procedures, but our procedures from there on in. So we've got different alert level systems that we have to oscillate between if we do start to get community transmission and from international travellers. But what has really helped New Zealand has been really good border control. So people entering New Zealand have to go into managed isolation facilities for about 14 days. And they're tested before they come into the country and then after their arrival and before they leave the quarantine facilities. That's been our leakiest, I guess, part. So we have had a rare cases of community transmission occurring because of that. But in terms of vaccine rollout, we are bringing in a vaccine in a circumstance where we don't have active community transmission. So for us, it's a non-emergency situation. So it has been a little bit different from other countries. This means that we've been a bit slower in terms of getting the vaccine rollout. So at the moment, New Zealand has enough vaccine pre-purchased for the entire country, just using a single vaccine, so the Pfizer-BioNTech vaccine. And we've got 4 stages. So the first stage was our border workers, so people who are at that interface, then our high-risk frontline workers, high-risk individuals, and then finally, around July, it will be opened up to the general population. And from our perspective, I guess the biggest risks for us are the efficacy of the vaccine. And I was very heartened by the presentations that we've had so far that suggest that the vaccines, particularly the BioNTech vaccine, looks like it has very broad efficacy against variants that are arising, because that probably is the biggest concern for New Zealand, is achieving herd immunity so we can open our borders again and maintaining that herd immunity.
Thank you, Joanna. What about the Israel run?
Now we're good. Hello and good afternoon or morning, wherever you are. So indeed, I think the experience in Israel was quite teaching. You know, Israel has seen some of the highest peaks per capita in infection in 3 waves that were pretty fierce. The last one, the 3rd wave, was driven by B117 and was particularly fierce and could not really be controlled even with full lockdown. And so in this very dire situation, basically to the rescue came COVID-19 vaccines. And within about 6 weeks from the late phases of December 2020, we had 40% of our population vaccinated. And at that time, within those, after those 6 weeks, we still had a pretty high rate of infection. We had about 8,000 cases per day in a country of less than 10 million people. And at that point in time, and despite the fact that the disease wasn't a check, we decided to open the lockdown. And this was based on our real world data that we have continuously collected. Israel has a unique repository of health records and digital records that allow us to do real-time assessment. In this case, it was a full-fledged assessment of vaccine effectiveness in the real world. We knew that we had over 90% effectiveness at that time. So we made the decision to simply open the lockdown and begin a gradual, continuous steps of opening up the economy. And we said morbidity will go down. It has to, because we're continuing to vaccinate. And it did. I mean, despite the stagnated point in which we made the decision, from that point on, we have basically seen a 100-fold drop in infection rates from 10,000 per day to less than 100. For 2 weeks now, we have less than 100 cases per day. We've went down from 150 severe cases per day to a single-digit number. And this last week, we had 2 days with 0 deaths. So we reached by now about 60% of our population fully vaccinated. And we basically, I think, hit pretty much the glass ceiling of willingness of the population to get vaccinated. So what we have left are the kids and 900,000 people who are pretty reluctant to be vaccinated. But at this point, there's enough indirect protection conferred by the vaccine to allow us to continuously open up. And right now, everything is open. Restaurants are open, schools are open, and we still don't see disease dissemination resurgence. But, you know, we keep our eyes open, and I guess we'll talk today also about variants and about other potential risks. But at this point, it seems the vaccines definitely do the trick, and we continuously monitor vaccine effectiveness. And it was actually higher in our assessment last week than it was 2 months ago, with 96% vaccine effectiveness in the real world in preventing infection, as was published in the New England.
Fantastic. Let me take the cue here to talk a little bit about variants with Esther Sabino, who is an associate professor at the Department of Infectious Diseases in the University of São Paulo in Brazil, and with Penny Moore, who is an associate professor at the University of Witwatersrand in South Africa. And both of you have been working for a while now trying to understand the emergence of new variants of the SARS-CoV-2 and how that impacts both, antibody responses in convalescent people and in vaccinees. So I was wondering if you could comment from your perspective now, how do you assess the danger posed by the emergence of these variants versus the speed that we are rolling out vaccinations, worldwide? So maybe, Esther, could you start?
Yeah, sure. So Brazil did a totally different epidemic, very tragic. And the first phase, phase, we had like a plateau. It took 4 or 5 months to the numbers to decline. And in the first phase of the epidemic, we had around 2 main strains responsible for the majority of the cases. In August, the numbers start to decline, and the government lifted most of the non-pharmaceutical interventions, although the number were still high. But 3 months later, we saw an increase in the number of cases. This is coincide with the local election. And we know now that early phase of in October, November, there was already a new variant that is known as P2, that has 1 mutation in the 484 position. And this was increasing all over Brazil until what we saw in Manaus, that was completely changed, a new variant that probably started in November, end of November, and in less than 7 weeks took over Manaus, and then slowly it spread all over Brazil. And today it's the main, so it's displaced the P2 and is the main variant in the country. And I think it's important that this new variant started in a region that had a high attack rate in the first wave. So somehow select or only a variant like that could repeat the same epidemic in a region like Manaus. And all this is happening at the same time as we are vaccinating. So we have now 30 million people who are vaccinated. 2 vaccines are available in Brazil. That's the Sinovac and the AstraZeneca. Somehow the initial studies suggest that they are being efficient in decrease the total number of cases, especially in healthcare workers. This is where the studies are being mainly done. So, but we are worried that with the vaccination and all the cases that we are still facing, we could again select, as it was the case in Manaus. So we need to be really paying a lot of attention to the variants that might overcome the vaccination.
Let me stop you there for a second. Penny, what do you think about the emergence of new variants, and how would you assess the threat that they pose?
Yeah, I mean, we're trying to weigh this very much in South Africa. So we've had 2 waves. The first of which was almost entirely dominated by 501YB2, the 5B.1.351. That second wave was much worse in South Africa, but the first wave was perhaps controlled by a very, very draconian lockdown. And it's, it's very hard to disentangle how much of the fact that second wave was so bad was due to overburdened healthcare systems, or perhaps likely due to increased transmissibility. The neutralization data all supports the fact that we should be seeing high levels of reinfection, but again, very hard to quantify in a system where it's very hard even to identify people who are infected and where most people are out in the communities. And I guess where we fortuitously benefited from the fact that we identified the variant in South Africa at exactly the same time that we were conducting 2 vaccine trials, one for AstraZeneca and one for J&J. And so totally by accident, rather than by design, we got some efficacy data for those 2 vaccines against the variant in South Africa. In the case of the AstraZeneca vaccine, the data that we had from a very, very small trial that was never powered to measure severe disease resulted in the suspension of the AstraZeneca rollout in South Africa, which was devastating because it was the only vaccine at the time that we had access to. And we're still playing catch up in South Africa from that. We have only vaccinated a couple of 100,000 people, only healthcare workers, with the Johnson & Johnson vaccine. And so, I think a combination of those 2 factors has really set us back in South Africa enormously. 5.1-YV2 is responsible now for more than 95% of infections in our country. It's spreading into every country where our sequencing is being conducted in sub-Saharan Africa. Many of those places are dealing with the same problems with vaccine access that we are. So we're very conscious there's huge public concern about the efficacy of the vaccines that we have and may get, perhaps if one day, against variants. But I think also large concern about the fact that we have a huge, huge population of HIV positive people in South Africa. So we fear that that this particular variant, in fact, we're seeing it already, that this particular variant is not going to be the first that we're going to detect in South Africa. So significant concern about what's still to come.
Thank you. And let me hand over to Nadia now to continue the discussion.
Thanks, Joao. I'd also like to welcome all our panelists today and thank the organizers for inviting me to moderate. With round 2, I would like to expand the discussion that we've started today and ask our panelists what are the most immunological questions to be addressed to understand, manage, and treat COVID-19. I'd like to start with Alan Fisher. Alan is a pediatrician, immunologist, expert in genetics of immunological disorders, as well as the head of the French government's Vaccination Advisory Board. Alan, my first question to you is linked to Shane's explanation today that severe COVID-19 has likely to do with delays or failure of triggering early innate immune responses. And Alberto has touched upon this topic as well. And I'd like that we expand this discussion. We've already seen immune signatures in patients with severe COVID-19, including children. And we already know some of the mechanisms involved in COVID-19 immunopathology, for example, type 1 interferon pathway. How can we use this knowledge to improve treatment of patients with severe disease?
Thank you, Nadia, and hello to everyone. Well, I think Alberto Mantovani already alluded to the discussion, so I can be short on this point. That is, obviously, there are several markers used as immune signatures for bad prognosis of COVID-19, and this includes, as you said, defective control of viral replication related to defects in type 1 interferon or delay in type 1 interferon production, as well as obviously as the many markers indicating immunopathology, excessive innate immune responses. As you know, there are hundreds of markers that could potentially be used on both groups of indicators of the disease. To me, there are two points that makes the things a little bit difficult to be applied on the daily care of patients. The one is that many of these markers are actually detected at a relatively late stage of the disease, at a time point where treatment might not be useful. For instance, an obvious thing is interferon. It has been clearly shown in mice and probably to some extent in humans that treatment by interferon relatively late in the disease will be detrimental, whereas perhaps not demonstrated, but it might be that early injection or inhalation of interferon might be useful. But how to detect these people at such an early stage, that is difficult because I'm not sure today we have the right markers. And also the markers we have are more designed to at the populational level, whereas at the individual level, again, it's very difficult to use them. So perhaps one of them that might be used, at least for some patients, is the detection of autoantibodies against type 1 interferon. because if those are present, from Casanova's work, it looks to be something like 13% of patients. Perhaps beta interferon can be given to these patients since they are anti-alpha interferon autoantibodies. But again, this should be given early. So I'm afraid I'm not sure I can provide you with a satisfactory answer to your question at this stage.
I have another short question for you. As you're a pediatrician and worked a lot of children samples, are vaccine safe for children?
Good question. Should we vaccinate the children? Well, there might be an individual benefit, not a big one, of course, because children do not suffer often from a bad COVID-19, but there is the multi-system inflammatory syndrome, which is not exceptional. In France, there has been 500 cases in 1 year. If we could prevent this complication, of course, it won't be bad. And obviously, the children do participate potentially to herd immunity. So they are a significant fraction of the population. So there is the temptation to vaccinate children. But obviously, the next key question, and the key question, is the safety. So how safe are the vaccines? There is this alert coming these days from Israel about the occurrence in some young adults of myocarditis. Is it a risk in children? So these issues have to be solved before considering to vaccinate children.
Thank you, Alain. I'll move on to our next speaker, Kylie Quinn. Kylie is an immunologist, vaccinologist, and research fellow at University in Melbourne, where she heads the Aging and Immunotherapies group. So Kylie, given that you're immune senescence expert, I'd like you to comment on age effect on immune responses against SARS-CoV-2. We've seen also from Shane's presentation that in elderly T cell responses are poorly coordinated and limited. They have a limited naive T cell repertoire. Can you please comment on this further?
Sure. So I think it's over the course of the pandemic, it's become abundantly clear that older people are disproportionately impacted with severe disease with COVID-19. And so when we're looking at a lot of those correlates of severe disease, things that pop out are things like we have decreased and delayed T cell and B cell responses. We have decreased and delayed innate responses, particularly delayed type 1 interferon responses. And then we have this hyperactivation of inflammation. And as Shane described, we see this lack of coordination between the different arms of the immune system. And when you work in aging and immunity, those are very familiar features. You know, those are real defining features of immunosenescence. And so it seems like there's these overlapping underlying mechanisms where some of the features of immune aging are similarly being induced in a heterogeneous way and perhaps a stochastic way at times in other settings as well. So aging is certainly a feature of this particular disease and severe disease.
And Kylie, how can we improve disease outcomes and vaccination strategies in elderly, knowing all these limitations?
Yeah, so it's a good question. So, yeah, so I think in terms of improving disease outcomes, of course, you need to prioritize better treatments. There's potentially roles for monoclonal antibodies there because there will always be a proportion of our population, particularly older individuals and potentially frailer individuals who we're unable to induce effective protective immunity in. In terms of improving the vaccine-induced immune responses, I guess I should note that the responses that we are seeing and the protective efficacies that we're seeing in older individuals is a great surprise. Well, I'm very positive about it. I think I was quite seeing the severe impacts and the disproportionate impacts of this disease on older people early on in the pandemic. I was really concerned as just what we were going to be able to achieve with a vaccine. But these vaccines are performing really well in terms of getting older people over the threshold of protective immunity. But there's clearly still areas that we can optimize them. So particularly if we're just looking at single doses of some of these vaccines, we can see stratification in terms of age having an impact in the magnitude of these responses. And so I think there's gains to be made there. Sometimes those gains can be made simply by boosting older individuals. So perhaps, COVID-19 may become more of an endemic infection in the older populations that we need to manage with effective boosting strategies. But there's clearly a lot more work to be done in terms of understanding the longevity of responses as well in older individuals, and that's a big question.
Thank you, Kylie. I'll move on to my next question to Florian Kramer. Florian is a professor of vaccinology at the Department of Microbiology at Icahn School of Medicine at Mount Sinai, working on development of influenza virus vaccines. Florian, we've seen today that different arms of adaptive immunity contribute to protection against SARS-CoV-2, and that these responses are quite heterogeneous. In your opinion, how far are we in our understanding correlates of protection in COVID-19?
Well, I mean, as Shane has shown earlier, there's many different mechanisms of the adaptive immune response that might contribute to protection. It goes from neutralizing antibodies to non-neutralizing antibodies to memory B cells to different subsets of T cells. And so it will take a long time to really figure out the exact interplay, and it might be different in different patient cohorts. But what we know by now, and there's relatively solid data about that, is that antibodies certainly provide protection. This comes through with the studies that you have seen by Davide Corti, where monoclonal antibody therapeutics actually protect from progression to severe disease. This has been shown by Dan Baruch's group by passive transfer of antibody into naive animals and then challenge, and this was sufficient to protect the animals. And of course, there are now also nice studies that correlate vaccine efficacy with neutralizing antibody titers. And of course, as you know, antibodies are easy to measure. T cells are much more complex, and so we tend to focus on that. And those are really interesting questions that we have to look at, but there's a more pragmatic question here. We're talking about correlates of protection. We are in a situation right now where we don't have enough vaccines, and the vaccines that are currently produced and which are licensed, the production rate is not high enough to cover the globe. And a lot of countries are struggling to get access to vaccines. Now it's getting harder and harder to do field efficacy trials, phase 3 trials with the vaccine candidates that are coming. And the more vaccine candidates we have, the more vaccines we have, the more supply we have. Now, it would be very important to facilitate the licensure of these vaccines to have an absolute correlate of protection. And this would be easiest with an antibody titer. An absolute correlate means that you have a certain threshold, and if you determine that the vaccine reaches that threshold or goes above that, you induce protection in an individual without actually looking if that person is now protected or not. This would be very, very important, and this would be very, very helpful in the current situation. And I think this should be the focus until we have enough vaccine supply. This is helped now by having an international standard, at least for antibody responses, because initially many different assays were used in many different studies, and comparing them across was hard, which doesn't impact on identifying a correlate, but it impacts negatively on identifying an absolute correlate. So I think this is what we should focus on right now, but academically, the interplay between different arms of the adaptive immune responses is of course much more interesting. And over time, we will hopefully disentangle all of this and see how this works out in different patient populations.
Thank you, Florian. I hope so too. I would like to move on to my last question to Adrian Hill. Adrian is director of the Jenner Institute at Oxford University. He's part of the team that developed, in collaboration with AstraZeneca, the Oxford COVID-19 vaccine. Adrian, I'd like to ask you, how can the current knowledge about immune responses against SARS-CoV-2 help us with future vaccine design for sensitive groups? Kylie has touched upon a little bit how this could work for the elderly, but maybe you can comment on immunosuppressed or autoimmunity patients and what are the key things to consider.
Sure, thanks. You left me with the toughest question to last. Thank you. Clearly, there is a challenge in many vulnerable groups, as you say. Interestingly, just to come back on your recent conversation, probably not in the elderly. If you actually look in the over 70s and even the over 80s in the clinical trials that have been done, it's remarkable how similar the antibody levels that we just heard about, and indeed the T cell levels are for most vaccines, RNA and viral vectors, in older populations compared to 25-year-olds. So that's a good starting point. Doesn't explain why the elderly are more vulnerable in general to disease, of course, but it's not a failure of those vaccines. Look, the real answer to your question is we need to know more about correlates, as you touched on earlier as well. And frankly, the clearest way of getting a correlate is to find a vaccine that works well and only induces one type of immune response. So if you believe in neutralizing antibodies, as I do, and I think most people do, having a vaccine that only does that and gives you antibodies and no effector T cells is very useful. Now, we completely fail on that because we use viral vectors, which are great ways of getting Cd8 T cells as well as antibodies. We get both. It's tough to pull them apart and see which is primary, but those correlate analyses are going on in very large numbers, and there should be more data soon. You might get more information from inactivated virus vaccines, Chinese vaccines and so on, where there are very few Cd8 T cells induced, or even traditional subunit vaccines in adjuvants. And sadly, they've taken a very long time to come along. So we are not going to have correlates on those for a while, if at all. I'm thinking of the GSK Sanofi vaccine and so on. So we're rather stuck with viral vectors, which are the largest number of COVID vaccines that have been licensed around the world, the most widely used. Ours, for example, is used in 130 countries as of last week, and RNA vaccines, which have come from nowhere to be fantastically interesting. Probably not more effective when you compare them head to head, say in surveillance studies. They're identical in their efficacy after a single dose, which tells you there's something odd going on between trials in the US and in other countries where all vaccines get higher efficacy in those US trials, including ours. So I think, you know, you're, as I say, if subpopulations, vulnerable populations differ dramatically, whether they're HIV positive, and use looks quite good in HIV positive individuals, immune responses look not very different, to late stage cancer patients where immune responses look a lot weaker. And that is the group I think we're particularly concerned about at the moment. Children are looking fine. I think they're going to be okay. They might not even need to be vaccinated. So I think it's going to be case by case in those immunosuppressed late stage cancer patients. The cop-out is to give them monoclonals, and if the numbers are small enough, then long-acting, albeit rather expensive monoclonals might be what you would end up using. But let me throw in one other thing just to confuse the pitch, because we haven't heard much about it, but we did hear a little bit from Shane, which is mucosal immunity. We would really like to protect young people so that we are protected from them. I speak as an older person. Young people transmit virus, they're not very sick. You could even argue that getting a mild infection is good for them. But to protect that population, you really need to protect the upper respiratory tract. And that is what all vaccines at the moment are least good at doing. Fantastic against severe disease, intermediate against mild disease, least good, maybe only 50% protective against asymptomatic infection. How are we going to deal with that? Well, several groups, including our own, are using viral vectors to give the vaccine intranasally rather than intramuscularly. That may well have advantages of getting higher levels of IgA in mucosal surfaces, particularly in the nasopharynx. A lower dose may be possible. It may well be easier to, which means you have more doses for the world, of course. It may be easier to deliver in mass campaigns, as we will hopefully find out. And it might even be safer in terms of clots and anaphylaxis and other things that other vaccines have had issues with. So I thought we should throw that in a discussion of immunology, that inducing mucosal surface IgA might be very good against this disease. We don't know at the moment. Thanks.
Thank you, Adrian. That's a very interesting conversation. I would love to speak more, but we have to move on to the next round. And I hand over to Seth. Thanks, everyone.
Thanks, Nadia. One of the many remarkable outcomes of this pandemic has been the unprecedented speed with which several viable vaccine candidates have been developed, tested, and disseminated in the general public. With good reason, my journal, Science, in December selected this inspiring and truly global effort as its breakthrough of the year under the heading Shots of Hope. I want to ask the panel about the technological advances that are transforming vaccinology. How much headway has been made over the past year? What are the obstacles currently facing us as this effort continues? And what comes next? I'd like to start by asking Penny, what are the key technological challenges to increasing global access to these vaccines?
So I realize it's Immunology Day and I should be focusing on immunology, but I think a lot of those challenges are actually fairly simple non-immunological things, particularly in middle-income countries. I think it's probably relatively low-tech improvements in the absence of stability of the cold chain and distribution networks and improved access and design and distribution of really simple things like better needles. I think manufacturing is a major problem. In South Africa, we do not currently have much capacity to manufacture anything, and we should, and so should many other African countries. So I think manufacture and the very scary IP issues that go along with that should be a major focus for us. I do think, I mean, the technological advances have been remarkable. And if you think about this in the absence of what I hope one day we will get a pan-reactive coronavirus vaccine, but if you imagine the possibility that we have to deal with variants going forwards, and then I think one of the major things will be how quickly we can tweak those variants. And I think that's pretty obvious in some of the platforms. Very fast is the answer. But I think there are follow-up questions to that, which is how quickly do you define how well that tweaked vaccine is going to work? And doing clinical trials is not a viable solution. I think that goes back to what Florian was saying about absolute immune correlates. We need to have a yes/no bar about whether our vaccines are doing well enough to protect against variants, and we need to be really fast in determining that. So I think that will be essential. I think what the other thing that the variants have taught us is that potency is king. It's the more potent vaccines that have done best against the variants. And so that brings us an interesting balancing act. Ideally, we would want single dose vaccines to maximize coverage and get them out to as many people as possible. But that's not how we get potent responses. So I think we're going to have to think hard about how we get really potent responses, because those are the responses that tend to be able to deal with the variants, albeit at a reduced level, but they still manage to do that. And I think it's been alluded to already. I think we need to think about ways to harness the fact that in places like Brazil and in South Africa and many other places, there's a huge level of prior infection that we can harness, and it's linked to this issue of a single dose vaccine, of getting better at working out who's been infected and giving them the single dose vaccine that gets them the potency that protects them against the variants. So not very complex technological things, pretty basic questions. But
Thank you, Penny. Florian. Oh, sorry, go on. Hello?
Yeah, we hear you.
Oh, okay, sorry. Florian, could you speak a bit about the concept of heterologous vaccination and tell us whether you think it might be a promising approach?
Yes, of course. I mean, heterologous vaccination, meaning combining platforms, giving platform A first, followed by platform B, is something that has been done in research for a long time. We know from HIV trials, we know from influenza vaccine trials that this can work wonderfully and often works better than giving the same vaccine platform twice. Now, so far, there's only a very small number of trials that are testing that. One is taking place in the UK for SARS-CoV-2. And of course, we need to figure out how the immune responses look. The prediction is that the immune responses will actually look better than giving the same vaccine twice. And the other important key question is, are there any safety signals? because there could be very rare safety signals that come with these mixed vaccination regimens. The truth is, several European countries are doing that experiment at this point in time. Germany, for example, has changed their regulations, their guidelines for vaccinating females, I think below the age of 60 with AstraZeneca. Many of them were vaccinated. They will now receive a boost with an mRNA vaccine. And of course, they will be tracked and we'll see if there are side effects. So in a way, this is getting tested. And on the other side, we have, of course, already many cases of people who got one vaccine in one country, then traveled, got another vaccine. So the expectation is that this could be an advantage and it could be implemented over the long run, but we need to see how the safety data looks like.
Thank you, Florian. Joanna, how feasible is a pan-coronavirus or a pan-beta coronavirus vaccine within the next few years?
I think given the leaps and bounds that we've made or that have been made over the past 18 months, it feels like almost anything could be possible. But this is a big issue because obviously there are many different coronaviruses, and I think it's reasonable to think that the coronaviruses coronaviruses with the ability to cause a pandemic will keep arising. So this is a really important issue. And a lot of people are working on the idea of developing a beta coronavirus vaccine or even a pan coronavirus vaccine that could be effective. I think and we know obviously, I mean, we had the David was talking today about identifying broadly neutralizing antibodies, so it is possible to develop these. I guess there are some important things that we need to understand and we don't necessarily know yet, and one of those would be how long lasting the efficacy of these vaccines that we have at the moment, particularly with the novel platforms like RNA vaccines, how long lasting those effects are. So they're all important considerations, but I mean, I'm very optimistic, and I think a lot of other researchers are very optimistic that that is an achievable thing.
Thank you, Joanna. Finally, Adrian, how will the advances in vaccine technology that have been spurred by this pandemic affect immunization programs for other current and emerging pathogens?
Yeah, I guess in several ways. My first hope is that they speed them up. Those of us who've worked on TB vaccines and malaria vaccines for more decades than we care to remember would like people to understand that there are millions of people dying from diseases like that around the world, and the pace of investment cumulatively in all of those diseases over the decades has probably been less than we spent in a year on COVID. You know, 1.5 million people died of TB last year. Hundreds of thousands of African children are dying every year of lack of malaria vaccines. We can make malaria vaccines. We just need to move a bit faster. So there's huge potential for doing good on the big killers. There's also a strategy to be devised that we could spend weeks on being better prepared for the next pandemic using technologies. But I think the biggest failing in this vaccine response to COVID-19 has not been in vaccine technology. They've performed really well. It's not been in speed of response, which has been extraordinary. It's been in lack of physical locations to manufacture vaccines, particularly widely around the world. But I don't mean, you know, in poor countries. Most of the big manufacturers are not in Europe, they're not in the Americas, they're in Asia, particularly in India. We need to sort out manufacturing capacity for the world. We need to have money to stand by facilities so that they can flip overnight to making outbreak pathogen vaccines of a defined number of types. We've learned what the better platforms are, at least circa 2020. We can focus on those and plan to move forward with them. So I think there's a huge amount to be done, but even to an immunology audience, we should be thinking about vaccination manufacturing.
Thank you, Adrian. I will now pass the baton over to Mia. Take it away, Mia.
Thanks very much, Steve. In this last 15 minutes, we're going to look a little bit at lessons that countries can learn from each other when it comes to getting people to actually take vaccines. And I'd like to start with Ran Balizer, who is the chair of the advisory team to the Israel government. We've heard that Israel, we know that you're the world leader in getting your population vaccinated. But as vaccinations progressed, your country did experience at least some vaccine hesitancy. You've mentioned earlier that you're struggling to get 900,000 people, which is about 1/10 of your population, to get them vaccinated and take vaccines. I'd like to know, how are you going to get them vaccinated? How are you going to convince them to take vaccinations? And are there lessons that other countries can learn from this strategy from Israel, seeing that you are ahead of all of us in vaccinating your people?
Thank you, Mia, for this question. I think that regarding those last 10%, I hardly doubt if we will vaccinate most of them. I think that we've reached the hardcore of vaccine hesitants and anti-vaxxers and additional people that I guess it will be very difficult to get, if nothing until now has made them understand the vaccine is both safe, effective, and is needed. Now that the disease is all but gone from the country, I think their likelihood of achieving that now is very small. But I would like to share with other countries that are now in the midst of the vaccination campaign some of the earlier lessons learned that we've had on what helped us. And one thing that we've noticed was a big discrepancy in vaccine uptake between different populations. And we saw that some segments of the population were not had higher rates of hesitancy at the beginning, and we knew that we had to focus on them. So these were some of the ultra-Orthodox religious Jews and some of the Arab Israelis and some additional subgroups that just were a little more reluctant than the others for the vaccine uptakes. And what we've done is we've done a lot of work with the specific community and especially with community leaders. We've sat with rabbis and with qadis and with local mayors and with leaders of local communities and with a lot of patients and a lot of proofs using local data. So we used a lot of local data to prove both effectiveness and safety of the vaccines. And once the community leaders were persuaded, the rest of the community, to the largest extent, followed them. And so in the beginning, there was really a problem of inequity in vaccine uptake. And now this is all but gone, with even all of these communities that I've mentioned have higher than 80% coverage among the older population and very high rates among the younger as well. So I think that we should remember that you could reach a very high vaccine coverage in your population, even with the beginning when there's hesitancy, as long as you're transparent, say about what you know that you know, and about what you don't know that you do not know, just explain why this is the right choice, not just underselling the potential risks. And working with local community leaders will make the trick. Thank you.
Thank you very much, Ran. I'd like to move on to Italy and France and look at the regulations and strategies they use. And so I'm going to ask Elaine Fischer, who's the head of the French government's Vaccination Advisory Board, and Alberto Mantovani from Italy, who is an emeritus professor of pathology at Humanitas University in Milan. I'm going to ask you to respond to the issue of mandatory vaccinations. So Italy has made COVID vaccinations for healthcare workers mandatory, but France, where several routine vaccinations are mandatory, opted to not do the same for COVID-19 vaccinations. And mandatory vaccinations are, of course, a very controversial subject. And I'd like to know, what can we learn from the reasons and the outcomes of the decisions of France and Italy? Elaine, we can start with you.
Okay, so in France, it was decided about 4 or 5 years ago to make the vaccines for small children mandatory, because at that time it was observed that there was a decline in the rate of vaccination, and it was going down every year. And despite quite a number of efforts of persuasion, it didn't work. And given the fact that these vaccines have been used for decades, they were known to be safe, it was thought that was a right idea, and indeed it worked. Actually, I think the Italians have done more or less the same at the same time, and it worked very well. But now in the context of COVID, we had a little bit of discussion, as in Italy, about making mandatory the vaccination for healthcare workers. at the time where we thought there was a little bit of hesitation and the rate of vaccination was not going up very quickly, but actually, contrarily to what I mentioned for the young children 4 or 5 years ago, for the healthcare workers, with time, it became possible to persuade most of them to be vaccinated. Today, there are about 80% of them who are vaccinated. And actually what worked best was the discussion with their colleagues. I mean, nurses talking to vaccinated nurses were convinced by them to get vaccine. So actually, I don't think there is a general common rule about what should be done. We should adapt to specific situations, cultural differences, and the history of vaccination in different countries. So it happened for France today, for the healthcare workers. Apparently, we do not need it. Hopefully, this is going to stay the same. But it might have been the case that we should have done it. So I wouldn't claim that we are more smarter than the Italians in doing so. Just the circumstances were different. So, but I'm curious to hear from Alberto about the situation in Italy.
Mia, shall I?
Yes, absolutely.
Yeah, I mean, France and Italy are very close, and in terms of mandatory vaccination in children, we had the same, and been at the front line, and the Italian Society of Immunology was at front line at the time when mandatory vaccination was reintroduced in the country. I should say that we have had a measles epidemic, and I have seen children with, not seen personally, but close, with acute lymphoblastic leukemia die of measles. I mean, that's terrible. So mandatory vaccination was reintroduced. There has been a debate as to whether we should have that for healthcare workers in introduced COVID. Now, the key point is that we have had shortage of vaccines. In most clinical situations, we have managed to convince people. I mean, my institution is virtually all of us got our jabs. There have been very disturbing episodes of people in retirement homes, for instance, and spreading the virus. I think it's perhaps too early now to introduce mandatory vaccination. In principle, I would agree with having mandatory vaccination in my institution for healthcare workers. I think it's a moral duty, and if people don't get that message, they should be compelled to behave. It's unacceptable that a physician puts his or her patients at risk.
Thanks very much. My last question is going to focus on herd immunity, and I'd like to address that to Esther Sabino from Brazil, who is Associate Professor at the Department of Infectious Diseases at the University of Sao Paulo, and Kylie Quinn, who is an Adjunct Research Fellow in Biochemistry and Molecular Biology at Monash University. So in my own country, South Africa, and in many others, such as Brazil, new variants are limiting our COVID vaccine choices, and we've also not entirely established whether vaccines protect us from getting infected with SARS-CoV-2, or for how long that protection that they give us against falling ill with COVID will last. So I'd like to know, will we ever reach herd immunity, especially if vaccines don't protect 100% against getting infected? We can start off with Esther, sorry.
So, yeah, I think Manaus reached such a high level of attack rate suggests that this is going to be very difficult unless we are able to mount a better immune response to the natural infection. So far, if things were left If we could not have access to vaccines, then probably herd immunity would never be reached. And now we need to see what's going to happen with the vaccines in place. I think it's too hard to think what's going to happen. We don't have enough data to be sure after the vaccination how we're going to be able to decline the level of infection. I think Brazil, again, probably starting the vaccine in a point where you have so many people being infected will probably is the place to learn and try to take a good look on the new variants that might surge. So I think we need to study. It's hard to know a priori. We'll have to wait and get the data.
Thanks very much, Kylie. The final word comes from you.
Sure. So look, I thought that was a great summary from Esther. I think, as you say, Mia, there are so many known unknowns. You know, we don't know about the longevity of the response. We don't know what kind of impact these variants of concern are going to have. And as Esther said, you know, there are places around the world where we could be looking at whether we will be able to establish herd immunity. I guess I was reflecting on this and just thinking, you know, in my part of the world, in Australia, we have a very different motivation for achieving herd immunity. So at the moment, we don't have a lot of free access to travel. And so with our vaccine rollout, we're trying to, well, the implication is that perhaps if we can achieve a certain level of rollout, then we may be able to reintroduce international travel. And so in terms of motivating people to go out and get the vaccine, I think then becomes a challenge of communication, because at the moment, the people who are most motivated to go get the vaccine are the people who are most at risk. So thank goodness we have really high levels of vaccine acceptance and vaccine motivation expressed by older individuals in Australia. But I think, you know, we still have to deal with that hurdle that Ran was talking about, like the discussion, engaging people where they are, communicating to them effectively about what this vaccine means and what it means to them, in order to get those percentages that really matter in terms of getting it to a point of high level herd immunity. So a lot of challenges, but incorporating communication is critical.
Thanks, Kylie. This brings us to the end of our roundtable discussion, during which we looked at global COVID-19 challenges, including new variants and the need for technology and transparent communication, and also the lessons we've learned so far. I'm now going to hand back to our chairs, Faith Ossia and Andreas Radbruch, to close today's event.
Thank you so much, Mir. So it's now up to me to say a huge, huge thank you on behalf of all the participants. Thank you so much to all our speakers, our panelists, our moderators. Thank you for waking up so early and staying up so late. Some of you will collapse into bed, but it's so much appreciated. There's been so much appreciation in the chat, and really want to sincerely thank you, and really to thank our organizers. Federica and Miriam put in a lot to get this off the ground, and we just want to appreciate them, as well as our KIT hosts who have managed the whole event and coordinated all the sessions. Thank you so much. I'd like to thank my co-host Andres for his handling of helping me to chair the session and bring this to a successful close. We want to bring your attention to the upcoming congresses. Andres, can I give you a minute to say something about ECI?
I hope that it's hopefully the last purely virtual Congress before the International Congress next year. But this year, the European Congress of Immunology will be a virtual Congress. Nevertheless, I think it will be a great scientific event, not only with European speakers, an international board, and many host sessions, guest sessions, for example, with IUIS panel speakers. And we had actually the motto, opening doors to the world of immunology, and we still have it. Please help us to make it a successful event. Join it, contribute with abstracts. Abstract submission is still open, and attend this great event, the prequel actually of the next one, which Faith is organizing. So.
Yes, thank you. The next one we're most excited about, this one's going to be in Africa. The travel situation remains to be seen, but just put it in your calendars. This is the IUIS Triennial Congress that was planned for Cape Town, and will still go ahead, and we wait to see whether there'll be some people in person or whether it will mostly be virtual. So with that, we've come to the end of our day today. Thank you for holding out. Thank you for contributing. Thank you to our speakers. Thank you to our organizers. And from me and Andreas and the team, goodbye. Goodbye.
And thank you to Federica, who had the idea to do this.
Yes, thank you, Federica and Miriam. And happy International Day of Immunology.